Okay, good morning. Today I'm going to talk about how to make the cells move. There are many ways in CompuCell to make it. Yes.
Gilberto, I'm going to ask you to restart. Please give the name of the module and also remind people that you're being recorded. That's a legal requirement that you do that.
Okay. Okay. Today I'm going to talk about the module 2.4 that was supposed to be given yesterday, and the workshop is being live stream, recorded and distributed, and you can find the material for this presentation on the Google Drive and that address that is in the screen. Okay. So the title of the talk is "The lecture is Cell Movement and the External Force." We are going to cover in this lecture these talks. I will present you the motivation for this kind of study and how to implement some important characteristic of the cell, which is the persistence in the movement. How to implement this persistent movement in the CompuCell with this special plugin called external potential, and we are going to make 3 exercises to apply this external potential to the simulations. Let's start with this motivation. The big question is, how do living cells, isolated or collectively, move around within their environment? I mean, why we want to study these things? Well, because you know that nothing happens in our body, in nature, in the beginning of every process, cellular process, that cell movement isn't present. For example, since the beginning of life, when you start your fertilized egg start to transform itself in a human being or any kind of living being, we have processes such as blastulation here, where the cells moving around to create different shapes and different tissues and lots of different structures during this process. We also have wound healing, where if you have a scar, the cells move into that scar and try to close it and regenerate the tissue, the skin, for example. Inflammatory response, when you have some infection on your body, cells have to move to that place of the infection or chase some strange organism in your body to chase them. Cancer metastasis, and it's another example of migration of the cells. The cells move from the cancer, from this region here, enter in the blood vessels and go to another place of the body, get out of the blood vessel and start a new source of cancer. So there are many pathologies also that cell migration is involved. So we need to study this kind of moment. The cells move with different mechanisms. These mechanisms can be external ones. For example, here you have a flagella movement, where the cell has a tail, and this tail ruffles and the cell moves. And in the other example, you have a paramecium animal, which has its movement through cilia around the body. So, but we have also internal mechanisms. And those internal mechanisms are built inside of the cell. For example, we have this lobopodial movement where the cell makes some protrusions in front of the direction of the motion, the movement, and all the body moves in that direction. Another kind of motion is the amoeboid motion. Here you have an amoeba movement in motion. See, the whole cell moves like a blob. Or we have also the lamellipodial motion, which is this one at the right movie. In this particular motion is the one we are going to pay attention from now. Okay, the lamellipodial motion. There is some, let's have a little look in the lamellipodial motion. But before that, we also have that. We also know that the cells have different behaviors. For example, this one that this fast movie I'm showing you, there are three kinds of movements. The diffusive one, the cells moving around but doesn't go too far. The persistence motion when the cell keeps going in some direction, and the mixed one when the cells diffuse and have persistence. Sure. Those kind of motions we can model in using CompuCell3D. How does motion occurs in the lamellipodium kind of motion? We have this engine inside the cell. It's a bunch of a big actin network that forms inside the cytoplasm, built from monomers of actin. These monomer actins in the leading edge of the cell here in the red part structure themselves in some fibers. Those fibers move ahead and make some little protrusions here, create some focal points. These focal points adhere to the substrate, and the cell is pulled back in that direction. The mechanism to form these actin fibers is like that. The monomers are recycled inside the cell, see, in a very particular shape. This angle between branches here have always the same value, about 73 degrees, if I'm not wrong. And the monomers go to the leading edge, attached to the fiber, and at the back end they are detached from the fiber. Then you have this kind of motion here, where the monomers are attaching to the front and detaching from the end. And the cell moves in that direction. In this movie here, you can see the creation of these actin fibers leading the cell to move in the direction where the fibers are growing. See? Okay, but this is a very, very complicated process. Let's see what we have to take into account to make our model. Okay. So, to begin with, lamellipodial movement is characteristic of the eukaryotic cells. So then we have the following. These structures, like we saw in the previous movies, they have a kind of lifetime because they to produce the emotion in a certain direction, like you see in this small movie here. See, they produce part of the lamellipodium. The lamellipodium goes in that direction. So we have here a kind of persistent motion. Also, if you look at the whole movie, a long movie, longer than this one you have here, the the trajectory of the cell would be a random walk. If you look at the small time scales, it has a small persistent time. That persistent time may be last for 30 seconds at most, unless you have some external ingredient in making the cell move from outside your system, like a chemical field, for example. So how do we implement this persistent random walk in the cell? I mean, in general, by default, CompuCell3D is produce movements that are completely random because it's a statistical process in the Monte Carlo metropolis algorithm. So we don't have persistency in the basic CC3D motion. We have to impose this by an external function. How do you implement that then? This is implemented by forcing the cell to move in a given direction. And this forcing means that we have to introduce in the effective energy of the model you are building an extra term, because when you calculate differences in energies, you are calculating forces from physics. Force is minus the variation of the energy, the gradient of energy. So this we have to introduce something in the effective energy, a term like that. What this term is telling you, that the energy is F term here, which may be a constant or not term, which stands for the intensity of the applied external force. This n hat here is a unitary vector that points the direction of the force at the position y that the cell is occupying. I mean, you look at the center of mass of the cell. The center of mass occupies the position xi. So in that position, the force of the cell have this direction n. And delta x is the displacement of the cell. I mean, the cell is in some position xy, and the cell is moving to the position x final. Okay, so this dot stands for the scalar product between f and the vector displacement, because the scalar product gives you a scalar, which energy is. So if you introduce this term in the effective energy, we are forcing the cell to move in the direction of this displacement, starting from the point where it is right now. Okay, so the external potential then follows that expression, where we have a constraint as usual, lambda vec, which is a vector with 3 components, each one in the following the coordinate axis, like lambda vec x, y, and z. It's scalar delta x, which is the displacement of the cell. Destination is where the cell is, or going to. Source is where the cell is. Now, if you make this scalar product here, you have 3 terms: xx, xy, and vec x, vec y, vec z. So to implement the plugin in CompuCell, we have to provide these 3 components of the vector constraint lambda. Sure. Let's try to understand. So we have those situations here. Now we can say how the cell moves. The cell green here is in that position at the beginning of the step, let's say. The red one is the force that is being applied to the cell at this instant of time. And then the directions of this red vector here is given by this lambda vector X, Y, and Z that you provide in your code. The funny thing is, because of the minus sign in the expression, if you want to the cell goes to the either positive direction of an axis, you have to apply a negative direction, a negative energy in that direction, because of how the CompuCell works. You have this negative all the time. Okay. So let's do the exercise here to implement this plugin in a very simple mode. Okay, as yesterday, I will guide you to this exercise. You have to go to NanoHub or your desktop, open to edit, and start to build a new simulation from scratch. And you have a 10 by 10 single cell, and the external plugin acts over the cell, making a force. The components of this force, the lambda vec X and Y, have the values -20, and the cell features is the lambda volume for the cell is 10, and the target volume for the cell is 300. And they use boundary conditions. Why boundary conditions? Periodic boundary conditions, because since the cell is going to move, eventually it will reach the border of your domain. And if you don't have periodic boundary conditions, the cell will be stuck at the border. So then we use boundary condition to this periodic boundary condition for the cell to move around and don't stop anywhere in your domain. Okay, please go ahead and try to build this simulation. I will launch this poll, and I wait for a few minutes to you to start the wizard and build the simulation. See, you have to use the plugins contact, volume flex, and external potential plugins. Besides the center of mass one, which is always included in any simulation you build from scratch.
Gilberto, I have a question from Duika to relay. She says, how should I set the boundary conditions?
How to set the boundary conditions? At the very beginning of your wizard, there is a window, a small window, and just below the size of the grid, that when you establish the size of the grid, there is this choice to choose either no flux or periodic. Let's see if I didn't.
Which one did you choose on the exercise?
The periodic one. I thought it is written there. Use periodic boundary condition.
Okay, didn't see it. Thank you.
Okay, I think now you are more useful with the wizard, more used with the wizard, so I can go ahead and see here. Okay, when you create the wizard, there is this uniform initializer plugin at the bottom. I forgot to mention that. You could use either of one, either the blob initializer or the uniform initializer. It doesn't matter. And you have to create the cell. For example, here I choose to put the cell right in the center of the domain. See, this domain has 256 per side, so the half of that is 128. Since the cell has 10 by 10 voxels, you divided the 128, pulling the cells some voxels to the right and to the bottom. So the cell started to 120 and then ends in 129 in X and Y sites. Okay, so this is what you have to do, and then you have to move to the start function to adjust the parameters given in the problem, in the exercise. You write for the target volume of the cell 300, like here, and for lambda volume, you have 10. Make these modifications. And now we introduce this new information to the simulation, which are the lambda vac X and lambda vac Y that will produce the force to the cells, over the cells. These lines that you have to type it. And note that we are defining those lines in the start function in the step below. It's information from the beginning of the simulation. And it is constant here. Okay, now we have to run the simulation. Let's see. Run the simulation. See if everything is going, what's going on. When you run the simulation, you should see something like that. Does everybody see that? The cell moving? See, this is why we are using periodic boundary conditions. The cell goes in a straight line, and when it reaches the border of the, I don't know why the thing stopped. When it reaches of the corner of the domain, it disappears there and appears in the left bottom. It's like the cell were moving over a donut or a tire, if you want. It's a torus. Although you are seeing here, it's a plane representation. The cell is moving over a torus-like surface, because this side here is the border here is the same position that we have at the left side, and the top side is the same as the bottom side. So the cell moves like in a circular motion here, and in a circular motion here. So it's like it was moving over a donut or a tire. It's an infinite plane to move. A surface, infinite surface, if you want. Infinite domain. Okay? This is why we use periodic boundary conditions. We can let the cell move everywhere without stopping, because there are no fixed boundaries. Okay, so another thing is, see, the cell is moving in a diagonal trajectory. Why? Does anybody know the answer? Try to answer that. Why the movement is in the diagonal of the domain? No? No one? Oops. Knows the answer?
In the chat.
I'm looking at. Yes, this is the answer. Yes, because we have put at the beginning both lambda vectors equals in both directions, x and y. So the cell will move in the diagonal of your coordinate system. Okay, let's advance and go to the next part of the exercise, which is how do I accelerate the cell? I mean, how do I change the applied force? Okay. This is kind of tricky. I will lead you more fast. So to change the force, we have to define a time interval to that change sense. I mean, we can choose any time interval we want. Okay. But if you, to be more, to have the exercise more visible, well, let's say, let's choose a certain amount of Monte Carlo step, which is not one. I said, let's say that we are going to change this. Let's choose the time interval like 400 or 1,000 Monte Carlo step between changing the intensity of the applied force. So how do you do that? Okay, that's where you have to introduce in your code. In the beginning of the steppable, you define a variable, which is going to be global because it's outside of any class. Okay. And here I am choosing the value of 100 for this interval between changes in the force and applied force. And here is the location where I make things change. In which way? See, I wrote here this if condition. And the if condition is testing if the Monte Carlo step is divided by the time interval rest 0. That means if the MCS is an integer multiple of the time interval, then we promote these modifications in the applied forms. Okay, so this condition here, if Monte Carlo step percent time interval, means that if Monte Carlo step is multiple of time interval, say if you have time interval 100, each time that Monte Carlo step is 200, 500, 1,000 or something like that, it's a multiple, integer multiple of time interval. It enters in this block and it changes the lambda vec x and y with the proportional to the value of the Monte Carlo step. I mean, I'm putting here float of MCS divided by 100, just to have a small number all the time, because Monte Carlo step goes to 10,000 and there is, I always explain later about that. Another thing that you have to transform MCS in a real variable, because we are divided here using the operation of division, and if you don't divide here by don't transform in a real variable, you're gonna be in trouble sometimes. MCS is an integer variable. Okay, you have to in division you have to put some real variables to divide. So we in this way, each time that the code enter in this block, satisfy this condition, the lambda vec x and lambda vec y will be increased in modulus by the amount of Monte Carlo step. Please put this in your code.
Gilberto, while people are taking a minute to do that, we have a question in the chat about Karina says, I noticed that you can choose between the pixel-based and center of mass-based external plugin. Does it matter which one you choose, or can you decide to adapt the parameters to get the same physical behavior independent of the chosen option? And then Ajay had a follow-up to that with, can pixel-based, if force can be pixel-based, can it be applied to cell membranes or perimeter pixels?
Well, I have to try yet, but I think it can. Okay, maybe we should test later in an exercise, but the default from the external potential plugin is the pixel-based one. And I think you're going to find this answer better explained in the manual of the external potential in the CC3D website. There is the manual there where you have the difference between both entries. In general, I use always the pixel-based model because I didn't need to change. But maybe you should try later, okay? Advancing here. So now you run the simulation. Let's see how the cell change its motion. Don't forget to here, I don't know, here. Let's okay, I think everybody has run the simulation already. So let's see. what you should see. See, the cell starts moving slowly, and as time goes by, the cell starts to moving faster and faster, and so on. Okay, I didn't go too far in time because this is the thing we're gonna to see next. Now we want to see the dependence on the speed of the cell on the applied force. So let us do the following. To see this dependence, we are going to build a graph. Okay. I think I skipped one slide here. No, it's okay. So we have to calculate the cell speed. We need to measure the speed between two positions of the center of mass. Okay, but for now, let's do the following. At the start function, we are going to create those entries in the dictionary that we're going to call old X CM and old Y CM. Those old terms in both entries means that this is the previous position of the center of mass. Okay. And at the beginning, at the start function, the old position is the initial position of the simulation. So, I mean, I call the attribute, the property X cone and Y cone from the cell, and put these values in the dictionary old. Okay, and then I create also this plot window that we learned yesterday. I go to the to edit, and from the CC3D Python tab, scientific plots option, you put the setup plot window and give you the names you want, configure it the way it wants. We are going to measure, to plot the lambda vec in the axis, the y and the x axis, and the speed of the cell in the y-axis. I mean, we are going to plot cell, speed versus applied force. Please do these inclusions. See, this is in the start function, don't forget that.
Gilberto, we have a couple of people having issues with their cell not moving, so we may want to just take a second.
Oh, okay, let's see.
Pause and let them. So Daniel,
who is the first one? My cell is not moving. Okay, let's go back then to see what could be missing in the code here. But who is asking? Liliana, did the cell move at the first time before you changed the force?
Liliana is in a breakout room with Pedro.
Yeah, so even after changing it, it's not moving. So I did the first one and then it did not move, it was static. And then after adding all these codes, yeah, it still didn't move.
Okay, who are you going to the breakout room? Pedro, could you go there?
Well, let's take maybe 1 minute to look at this because other people may be having the same issue.
Okay.
Liliana and Pedro are currently in a breakout room, and if we can't get it fixed quickly, Daniel will send you over there.
Okay.
Okay.
Yeah.
Yeah, Louise has the same
So,
problem, so it's
yeah.
probably worth looking at as a group.
Does
I
somebody
think
want to
from
share
the-
your screen back, maybe, and we can look at it together?
Yeah.
So Gilberto, why don't you stop screen share and we'll look at this together for a minute.
Okay, I will stop screen share.
Okay, Daniel, why don't you screen share?
Okay. Yep. I don't know if you can see my screen.
Yes.
Yes.
Mhm. So there's my XML file, and then there's a steppable file.
So you have the explicit forces there.
Yep.
Where is the
No.
external potential plugin? I think you didn't load it. Ah, it's there. It's there.
Yeah, is there? Yeah, external potential is here.
Scroll down.
Ah, you didn't use the flex one. I forgot. I think that may be the reason.
You'll have to, I think, get rid of line 39, yes?
Yes.
I should get rid of line 39.
39 only. You get rid of it. Because we are going to define the lambda vec in the steppable.
Why don't you resize the,
Yeah.
so it fits in your,
Okay.
did you save the,
No.
did you save it?
Yeah.
Now it's saved because it's in blue.
Still.
Ah, it's
Oh yeah, it's moving. Yeah, it's
Yeah.
moving. All right. Yeah. Thank you.
Did anyone else have a problem with that? Let's make sure everyone can get this to work.
Louise, were you having the same issue?
Did you share the screen?
Yeah, if you'd like to share your screen, Louise, we can take a quick look. So
Okay,
to open Twedit.
Let's see the HTML. Yes. Here. Then
I'm missing something here in the external potential because I've commented that out,
yes.
but I couldn't work out where you said to put something.
And see,
That looks fine. Now go to the Python and see what the Python is.
it seems right.
So you haven't added the force generator in the Python.
Whereabouts do I need to put that?
You can put it there where you are in the step function.
Ah,
How do I do that?
Go to CC3D Python
Yeah.
and then let's see, which one is it, Gilberto? I never use this.
no. This external plugin doesn't have anything there. It is called, the plugin is called from the XML, and then in the start function you just have to add the lambda components.
Were people having trouble with cells not moving? Their cell's not moving.
Yes.
So I just had that happen, and I just had to delete the divided by 100.
Ah,
But Gilberto, you're going to have to screen share again to show the code that needs to be put into that
okay.
the Python.
Yes. And also, Gilberto, you have about half an hour left, just to let you know. Louisa, maybe we can help you in a breakout room if you're still having issues with your code.
See, this is the code. The time interval is defined here. The lambda components are defined in the start function. And the change in the step function.
So why don't we leave this up and make sure everyone has time to type it?
Okay.
Or do you have a version of this code that they can upload?
Yes. It's in the student. Let me find.
Don't worry about it. You work on that. I'll look for it. Okay.
It's in the demos and the 2022 folder, students material demos exercise 2.4.2.2, because I divide here to have different codes for each step of the simulation of the exercise.
I would like to add something. So if people still are not seeing cells moving, as Liliana just mentioned, the divided by 100 in lines 25 and 27 probably makes the force update too small. So if you decrease that 100 to 10 to 1, may help you see the cell moving, okay?
Oh, that maybe it helps.
Let's come back and make sure that everybody has this working.
I set that number to 20 and it worked really well.
Great. Okay, does everybody else getting it?
Yeah, probably it's yeah.
I still can't get mine moving at all. I've gone back and checked the code several times. I'm wondering if it's the way I've set it up or something, if I've missed a step.
Reduce that 100 divided by 100 in 25 and 27 lines inside the step function. Okay, step function inside the loop, you have the lambda vac x, lambda vac y. Reduce that divided by 100 to 10 or 20, like Liliana suggested, and test again.
I tried that. That didn't work either.
And didn't work?
No.
Okay,
Pedro,
so
there was also the thing we had to comment out in my HTML file.
okay, probably this is a problem as well. Gilberto,
What?
We showed
no, so enter a breakout room with me then. Breakout room, room 2. Okay, so people that need help, please join my breakout room, room 2. Okay, and I can help you there.
Trying to go there.
Yeah, so like Pedro said, if you're still having problems with getting your cell
Gilberto,
moving,
you
or
can't
with
go
your
to a
code,
breakout room. Yeah. Only
Um,
if anybody's having trouble, please go to breakout room 2.
and Gilberto, you can go ahead and continue, because I know we have some
Okay,
people
I'm
who do
going
have
to
this
continue,
room.
then you solve that. Okay, let's continue here to the next step, that is to produce the graph when we can see this cell changing with the value of the applied force, the speed change. So here is what how we have to calculate these things. This is a little trick. Why? Because, as I was saying before, we have to calculate the speed between two positions of the cell. So you know the position at one point, and then you know the position in the second point. You calculate the speed using the usual speed definition, delta delta space over delta time. Okay, so our delta time is the amount of Monte Carlo step. Sure. And how do you calculate the delta space? Well, you have to divide the motion in components as usual. You have here d delta x or dx, which is the actual position of the cell center of mass minus the old position of the cell center of mass, which is stored in the dictionary. Here you have to be careful with this periodic boundary condition. This is a very important thing when you use periodic boundary conditions, because when you are calculating these variations, since the cell moves from, for example, the cell reaches the border of the boundary, say x equals to x limit. When it moves to the left, enter by the left side of the domain, its position there is x equals to 0, for example. Then if you take this difference between 100, which is the right limit of the domain, minus 0, which is the new one, you have a difference of 100, which is not true because the cell just walked one voxel. He went from 100 to the right, from the right to 0 to the left. So the motion is only one voxel. We have to correct this defect by adding a term which takes into account this boundary condition. So you say if the distance between the difference between the distance is bigger than half of the dimension of the domain in that direction, we decrease from the dimension of the domain that amount of the cell movement. So you are correcting the steps by the size of the grid. So the delta, the variation, is the real variation of the cell. This is why you do not see the cell make some strange movements when it's passing from crossing one border and appear in another one, because we have this kind of correction in the CompuCell mechanism. We have to do this for both axes, X and Y. So doing that, correcting those distance, those variations in displacement, we can calculate the speed of the cell in the usual way. The speed is the square root of the sum of the components, the square of the components. It's a vector thing. It's a modulus of a vector divided by the time interval. So we have here distance over time. So it's the speed of the cell. Did you guys understand that? How we calculated this? The way to calculate speed is the usual way, distance over time. The only trouble here is to make the distance correct by taking into account these periodical conditions of the grid. So we have to calculate also the intensity of the lambda vec. Since it's a vector, so we this intensity, the modulus of the lambda vec is again the square root of the sum of the square components. Lambda vec square plus lambda vec y square is square root. And we add this, this point. Lambda vec is the x-axis of your plot, and the speed is the y-axis of your plot. That we add this data point in the graph. And finally, at the end of this loop here, we update the old dictionary entries with the actual position of the center of mass. So we have here the updating. What? Something talked? Okay, so we update the code, the dictionaries, and go off. Okay, try to make these modifications in your code to implement the graph. Okay, since I was talking a lot, I think everybody has included that in the code so far, so let's move on.
Does anybody need help, or is everybody ready to move on? Anybody needs help, please say so in the chat.
Let's see. Need time, okay. Why use abs dx? Because the cell, the displacement could be in the opposite way. I mean, the cell could move from the right side of the screen to the left side of the screen. And then your delta x would be negative, or so you always take into account the absolute value, and it works either way, if the cell is moving left or right or up or down. Okay, code again in the screen is that. Let's take out the. Let's see if there is more questions here. Self-dim. The question about self-dim. Self-dim, dim X is internal functionality of CompuCell that gives you the dimension X of your grid, of your domain. Is how do you call? Remember at the beginning of your wizard simulation, you define the size of the grid, 250 or 100, whatever, in the X, Y, and Z. And during the simulations, you can access those values, the dimension of your grid, with this self-dim X, self-dim Y, and self-dim Z. This gives you that values defined at the very beginning.
I have a doubt. So why do the cell starts diffusing and spreading when it goes to a higher velocity? What is causing that in CC3D?
because you increase the amount of voxels copies. When you decrease the energy, you increase the probability of changing the number of accepted copies. So the fluctuation of the I say membrane of the cell will be higher.
Okay, so do you think if I put a target surface with a high lambda, that won't happen? If I do not let the surface to become that diffuser?
Why are you
But-
concerned about it?
Well, just by the motion, I do not want my cell to, if it moves at high velocity, I do not want my cell to become spread. I do not want the spreading to be a function of my my velocity. Is there a way to remove that?
No. Fundamentally, the answer is no. You would have to rescale all of your other parameters.
I see. Okay.
If the cell is beginning to fall apart, and I think Gilberto will address this, then the force you're applying to it is too large.
Right,
Yeah.
right. It's probably that. I should not be applying that much force, but yeah. I was just wondering if you can do that.
But you could certainly increase the surface constraint and the other constraints
Right.
to be proportional. Yes. And that would
But that'll
reduce.
only prevent it to some extent, right?
But if you increase the other lambda, the other constraints, probably will prevent the cell to speed too much, the velocity increase, because you decrease the probability of copies.
Yeah. I get that. Yeah.
Okay, everybody finished? Oh, good question, Daria. That's the point. Okay, let's move on and show the results. Here's what you get. See? I took this from the image because I cannot wait too much to get to this point here, the plateau of the cell speed. And see, at the beginning, you have a big growth in the cell speed here. The derivative is very high here, but then the speed starts to stabilize. Doesn't matter how big is your force, the cell doesn't move faster. Okay, and why is that? Does anyone knows the answer? I'm waiting. Let's see if somebody finds the answer. No? Yeah. Okay, let's answer it. So the point is the Monte Carlo step, it makes your COPS attempts to choose the boundary of the cells only once. So as long as you increase the force, you increase the probability of changing the voxel copies, but the maximum that you can reach is the quantity of the cells that are in the border of in the membrane. And this membrane, each voxel of the membrane can be sorted by in the Monte Carlo step only once. So it doesn't matter how much force do you apply in the cell, the number of the voxels that the membrane can advance is at maximum 1 per Monte Carlo step. So it reaches the maximum with at some point of the applied force. There is no much change to the cell speed. Most of that amount of pixels voxels that it has in the membrane, because the inner parts of the cell doesn't matter for the voxel copy. They don't enter in the voxel copy. Only the membrane, and the membrane is limited, and it can at maximum run 1 pixel per Monte Carlo step. So this is why you have this plateau in the speed limit. Okay, so you have to figure out now from this kind of exercise what would be the good parameters to fit the simulation with your experiment, if you want. This is the most complicated thing in building simulations and comparing with experiments. How do you translate voxels in Monte Carlo step in nanometers or micrometers and microseconds? Okay, this is a very complicated problem. Okay, let's move on here. Now, let's see, we just built one cell, a single cell with no structure at all. It's a blob of voxels. But in real world, the things are much more complicated. I think the cells have a mechanism, a structure inside, and we can see this moving. We have the lamellipodium moving, we have the nucleus, we have the internal structures like mitochondria and other organelles inside the cell, the cytoplasm, the lamellipodium moving here. So basically, we can say that the cell has, at the beginning, 5 components, which is a model proposed by Abercrombie in the 70s. And this model says that let's consider a cell like having a nucleus, the cortex, the cytoplasm, and the lamellipodium. And the cell is moving over a substrate like that, the yellow part, and inside the middle. This is the thing we aim to attain. Okay, that's our model. And how do the cell moves in this model? Oh, it's well known. I mean, there is some protrusion here. The leading edge of the cell, the lamellipodium, move forward, attaches itself to the substrate, creating focal points here, focal contacts. Everything is pulled forward from the cell, detaches from the here part of the cell, detaches from the substrate, and is contracted in the same direction to the right side. And then things start over. So it's like that. Advance, attach, detach the here part, contract the here part, and move in that direction. So this is a single 4 steps motion here. Now we are going to build a kind of more, I call it the more realistic cell which is being formed, composed by 3 kinds of cell. We will have the cell type lamellipodium that I call lamel, cytoplasm, and nucleus. The problem, the biggest problem in this exercise is to adjust the cells energies and constraints to make the resulting cell in the simulation resemble as a real one. I mean, the nucleus must be inside this cyto, which is surrounded by lamel. This is the first part of the exercise. I think this one, it's complicated. So I will type it here and you follow me, okay? Let's open to edit here. And build the simulation from scratch. Okay, let's go here, project, new project. Let's say E2 Porf 2, no, exercise 2 for 2. This is the name of the simulation. Python and XML. Next. Now let's use this periodical boundary conditions. There is no third dimension, only 2D. Let's type square, everything remains the same. Next. So now we add the cell types. Let's call lamel, add cyto, add, and nucleus. Okay. No fields. And here we have contact, volume flex, and the external potential flex. The properties and behaviors are the very same as the previous simulation. No other thing. Next, finish. It's okay. Open here the files, and let's go to the XML file. In the XML file, we have to create the cells. Everything is here, the 3 components, the 3 cell types. Lamel, I wrote lamel in the lowercase. Okay, doesn't matter. It's just an exercise. So we have to create here in the steppable initializer, uniform initializer, 3 cells again. So now we are going to repeat this block. Control copy, Control V, 3 times. The first one will define the lamel cell, which is going to be positioned at the center of the 120. I should have a copy after 129. 120 and 29, 10. Here again, 120, same X, but different Y. To not be superimposed to the previous one. And this is going to be the site.
So, Gilberto, we are at 11:30.
Let
Since
me
we started, right? No, we started 10 minutes late. So just letting you know, we did hit 11:30.
Ah, okay.
So.
And here is 130. 139.
Yeah, I think the box marks X is 139, not 129 for the cyto cell.
Yes, in the same X position, but different Y position, the cyto.
Oh, okay. Okay.
And here the last one is in the same different X position, but the same Y position than the first. Okay. And then we have to go to the steppable file to create the cell volumes and lambdas to each of the cells. So far we have this, okay? We just did this part here. Now we have to go to the steppable here. In the start function, we are going to say the following. Well, now we have a 3-component cells. Let's do the following. Let's be here. Let's see. We have to define a bigger cell because now our cell has more components. And each component has a different size. For example, we can say that the volume of the lamellipodium is 30% of the volume of the cytoplasm, and the nucleus is 20% of the cytoplasm volume. Okay, so we have to divide, to include in the simulation these volumes and constraints, which are the same here for all the components of the cell. So here in the start function, we make a loop, visit all cells here, okay, and say, let's do a little bit different. Cell dot lambda volume equals to 10 because every cell has the same lambda volume, and then we test if cell dot type equals to self dot cyto cell dot volume equals to something that I will call cell vol. And this value of cell vol, I'm gonna give here as a constant, 300. Okay. Then I have to define the size of the other components, control copy, and say, here's this lamel that I have to write everything in capital, because when I use self, I have to use capital, is, oh, it's not too 0.3 times cell wall. Here I made a mistake. And here nucleus. .2 times cell wall. I don't need this line here. Okay. This is the beginning of our cell. Let's see how it does it look like. Save it. Let's see if it is going to work. Open the player. Oops, what did I miss? Doing on the fly is very dangerous.
Should the if statement in the if should be lowercase?
Excuse me, I didn't understand. Should what?
The if statement, should it be lowercase or it doesn't matter? In the Python steppables file.
Let me see here where is the error. Here, line, let me check here, just a line.
You have capital I for if.
Ah, yes. Thanks, James.
I wasn't the one who caught it. It was our participant who caught it.
Okay, thanks, guy. So, see, here is the cell. We still have an error. We have 3 components here, but let me fix this error. Cell.volume equals to 90 is illegal.
Target volume, not volume.
Yes, I am trying to do fast because Hayden has warned me about the time.
One way to avoid this kind of problem is to use the twidit cell property menus.
Yeah.
So then you don't have to type things by hand.
Yes, yeah, you are right. And let's see if you made a mistake here. 2029, 3039, 2029. Okay, I think now we are good to go. Save it. No? Okay. See, these are the cell components. 3 components. 1 is lamel, 2 is cytosol, and 3 is nucleus. I think what was the mistake. I think I forgot here to say that the nucleus is 10.
Yeah, sorry, where did you define the cell wall?
Here at the beginning of the steppable start function here.
Oh, okay.
Okay, now it's no error. Okay, this is the initial step. We have 3 component cells in the center of the grid. If I put it to run, I hope it doesn't crash. We have a wrong cell because the energies are not correct adjusted. So what we do have to do now is to come here. This is a kind of time-consuming thing to do, is to try to adjust the energies here on the fly to get the right values for correcting the cell shape and structure. I think this nucleus is too big. I don't know why. But anyway, see here you can, for example, you want that the nucleus goes inside the cytoplasm. Then you must prevent the nucleus to be in contact with the medium. To do that, I came here, for example, in the nucleus part of the contacts. I see the interactions between nucleus and lamel, nucleus and medium, and nucleus and site. So here, everything has the same value. So it doesn't matter who is touching who here, because the energies are the same. So I have to adjust this. To prevent nucleus to be in contact with the medium, I have to increase this energy here. For example, to 40. See? It escaped from the medium. It goes to inside of everything. But it's still, we don't want also that the nucleus meet the lamel. So we have to avoid that. So look at the lamel here, lamel nucleus. See, it's 10. We have to increase this energy, because as bigger the energy is, the less things it likes to be in contact. Let's see, 25 here. See? The nucleus goes to the center. There is something wrong. I think that the nucleus is too big, but I will not lose time trying to fix that. It is in the exercise. The next thing is to make the lamel to likes more to cytoplasm, because the lamel must be spread around at the beginning around the cytoplasm, and it only takes this grouped shape if the cell is moving in one kind of direction. So we have to go to lamel and where is lamel and cyto here, and decrease the energy, because lamel likes cyto. So let's try 5 here. This I think is not enough. Oh, see? It's maybe 2. You know, this is a long work to do. Anyway, this is the way we adjust these parameters. And then I will give you the right ones that I spent some time doing yesterday. It's in here. Let me go on the presentation. I'm just going to finish this.
So Gilberto, we are at 11:45. We do need to move on to break so we can get ready for Pedro's talk. Is there something that you can give them maybe to work on a little bit over the break for those who are interested in finishing, because we are out of time for this session?
Okay, just the last slide is the contact that I have found to get the correct shapes for the cell, the structure of the cell. So you can copy from this, and it is in the exercise in the demos at the website workshop. Okay, then If you,
I will post that link in the chat here to the exercises. Let me just grab it.
Mhm. Okay, then I finish here. Thank
So please fill out the module. If people would like to go into a breakout room during the break with Gilberto,
Yes.
go over some of these things, why don't you use breakout room 2? But please fill out your module questionnaire first before you join the breakout. And otherwise, we'll see you all at noon to start the next session.
Okay, so I will stop to share
you,
my
everybody.
screen.
I have a small trouble. It's actually regarding TweetIt. I've somehow managed to get rid of my the left section of the TweetIt where you basically double click on CCD 3D files to open XML and Py. Is there a way which I can bring it back, the toolbar?
Yes, so please go with Gilberto into the breakout room and he can show
Okay,
you how to do it. Is there any other questions or need? AJ asks, how do you implement proliferation of multi-compartment cells? I don't know if we're going to cover that on Friday, but that's a good question. Ask Julio to show you how to do it if it's not covered in the class. There is a special mitosis steppable for compartmental cells. And again, please, everybody, do fill out the module experience questionnaire and then go get a cup of coffee and come back in 10 minutes. If you have questions, please join breakout room 2 with Gilberto and he'll help you out during the break. Give people 3 minutes to finish the module experience questionnaire.